All right, our final discussion in this unit is going to be about enzymes. And enzymes are a very special type of macromolecule, specifically a protein. And they are incredibly important in maintaining that balance of energy plus within a cell, and then the cell's ability to have enough energy to perform all of the tasks that it has to perform. And so our essential question for this segment is, what are enzymes and how do they function? So let's just start with an overview of enzymes in general. So chemical reactions are happening in your body all the time, hundreds of thousands every day. And these reactions are going to require energy. It's just there's no way of getting around that. But there is a very limited amount of energy in our bodies in order for all of these things to happen and be performed appropriately. And so the energy that's needed to get a reaction started, though, is called activation energy. Enzymes then are going to act on that activation energy. So specifically, enzymes are proteins that act as biological catalysts. They speed up chemical reactions that take place in our cells. And in order to do that, what they actually do is they lower the activation energy for a reaction to occur. And so if you look at the general graph there, you can see that with enzymes, the activation energy is much lower than it is without enzymes. And so you can actually get almost 3 reactions in for the same amount of energy that it would have taken for this chemical reaction to occur without an enzyme. So a catalyst is a substance that speeds up the rate of a chemical reaction. So our cells use catalysts to make chemical reactions happen at a lower energy, which allows more reactions to occur with the limited amount of energy that we have. Enzymes are easy to identify as we're reading different words, because generally enzymes will always end with the suffix ase or ace. So examples in our body: helicase, it is the enzyme that unravels DNA helix during replication. Amylase is in our saliva, and it turns starch into sugars. And then lipase, which breaks down fat molecules in our stomachs. So how do enzymes work? Well, enzymes provide a place where a substrate can be brought together to react. And substrates are the reactant molecules the enzymes act on or break apart. So if you look at the diagram there along the left, picture number 1 shows the enzyme and the substrate. Then you can see the substrate fits right in or clicks into place into the enzyme. Once it's there, the chemical reaction occurs. That's what's happening in picture 3. And then picture 4 shows you how that reaction is finished. The products leave the enzyme. And then the enzyme is actually available to be used over and over and over again. And so that's one of the great things about enzymes. Not only do they lower the activation energy, but they lower the energy and resources spent on rebuilding molecules because they are reusable. So when we're looking at enzymes, the location where the substrate binds to the enzyme is called the active site. And so you can see the active site in this case is marked. The green substrate clicks into place. Again, the enzyme begins the chemical reaction. Specifically in this case, the enzyme changes the shape of the substrate as it binds. When that occurs, then the substrate goes through its chemical reaction and the products are released, leaving that active site open to go through the reaction once more. The shape of a protein, remember, absolutely determines its function, and that's especially true in enzymes, because enzymes and substrates are referred to as a lock and a key because the shape of the key matters in order for it to open a lock, and therefore the shape of the enzyme matters in order for it to bind appropriately with the substrate. And what that means is that only certain substrates can fit into specific enzyme active sites. So remember that we keep showing enzymes as these cute little pictures with lots of these little specific shapes and things, but remember they're large proteins with lots of atoms that are chemically bound together, and so they're more of this big glob and not so much of these cute little specific lock and key pictures. So, can an enzyme work at any temperature? So looking at this graph, you can see that enzymes have an optimum temperature at which they function best. And so what would the temperature be that most enzymes would work best in our bodies? That's right, 98 degrees or 37 degrees Celsius. And if you look at the graph there, you can see that the enzyme is more or most active at 37 degrees Celsius. So how does pH affect the rate of enzymes? Well, enzymes have an optimal pH at which they function best. So each enzyme is going to function at a different pH depending on where it is in the body and what its function is. And if you look at the graph there to the left, you can see 3 different enzymes operating at 3 different pHs where they are most effective. The blue line being an acidic environment, the orange line being a neutral environment, and the green line being a basic environment. And so which of these enzymes would work best in your stomach? Would it be pepsin, which has an optimal pH of around 4? Would it be amylase, which has an optimum pH of 7? Or trypsin, which has an optimum pH of 8? It would be pepsin because pepsin operates in an acidic environment, which your stomach is. So how do pH and temperature really affect an enzyme? Well, heat and chemicals are going to break bonds between the amino acids, and they're going to cause the protein to change shape. And when an enzyme changes shape, it no longer works. We refer to that as denaturing. And so when an enzyme gets put into an environment it's not built to withstand, it denatures, and when it denatures, it stops functioning appropriately. So basically, it has been killed. So if we watch this little video, it will show that. Oops.
Extreme temperature, pH, and salt concentration can change or destroy the structure of a protein, and consequently change or destroy its activity. An egg provides a good example of how heat can change the structure of proteins. Proteins are found in both the egg white and yolk, and these proteins have different structures in raw versus cooked eggs. Let's consider the proteins in an egg white. In their native uncooked state, the proteins are chains of amino acids that fold into complex shapes. Several types of bonds form between amino acids in a chain, stabilizing the structure of the protein. When the egg is cooked, the heat makes the molecules vibrate. The bonds linking the amino acids in chains are strong covalent bonds that withstand the heat. However, most of the bonds that stabilize the three-dimensional structure of the protein are weak bonds which break more easily, allowing the proteins to unravel. The destruction of a protein's three-dimensional structure is known as denaturation. When 2 unfolded proteins make contact, they form new bonds with each other. A large net of interconnected proteins forms, and the egg hardens. Light can no longer penetrate the egg, which is why the clear portion of the egg turns white.
So go ahead, take a minute, and summarize your notes as you have learned them about enzymes.